And then population characteristics. I won't go into the details of all of those, but you have to understand anything that is different in a population can cause an infection to spread. You know, immunity levels, genders maybe, age, nourishment, all of these can play a role. We mentioned 3 types of studies using epidemiology. The descriptive study, this is the first study that comes out when you show up at a scene where there's been an outbreak and find out the details about a place and time of the outbreak, who was infected. Then there's an analytical study, one that actually does a more intensive, extensive, if you will, look at the population, a cross-sectional study of people to see who are the more relevant individuals at risk. And then that is generally followed up with an idea of where the study came, or where the disease came from, and where it might be going. So they do a retrospective look at the disease and a prospective look at the disease to see where it might be going. And then finally there are experimental studies when we've identified medicines that might be effective in treating the disease. Then we'll see experimental studies carried out. Three organizations, National Centers for Disease Control and Prevention, the CDC, the Public Health Department, and the World Health Organization. You can just imagine me asking you a multiple choice question. Which one of these health organizations is responsible for monitoring water at the local level? Or which one of these organizations puts out a weekly report on disease? Something like that. So know which one of these organizations has a role in countries outside of America. That's almost too easy. Nosocomial infections, know what they are, know how they are spread, know the reservoirs for nosocomial infections. So that brings us to antimicrobials. A couple of things that we want to keep in mind with regards to these. First, we're talking about chemicals now. Chemotherapeutic agents. Many of these are produced by bacteria. If you remember our discussion on some of the features of antimicrobials, one of the important things was their toxicity, specifically their selective toxicity. You know, it's ideal if they'll do greater harm to the microbe than they'll do to the host. And so with that in mind, we can talk about their therapeutic index then. And those microbes with those chemicals or antimicrobials with a high therapeutic index are not going to exhibit any kind of toxic effect to the patient because they don't target anything that the patient might have. But chemicals that have a low therapeutic index have to be monitored because they're probably targeting a molecule that both the bacteria has and we have as well. And so if the levels get too high, it can be detrimental to the host, and that's certainly going to be self-defeating. A couple of other things to keep in mind. The action of the chemical, is it a bacteriostatic or is it a bactericidal? Does it inhibit the growth or does it kill the microbes? See, penicillin doesn't kill the microbes. Penicillin inhibits the growth because it interferes with the formation of the cell wall. It's only effective in metabolizing microbes, actively growing microbes, because it's interfering with their ability then to grow. But if the microbe isn't metabolizing, then penicillin isn't going to kill it. It's not going to target and kill it. It's just going to interfere with its ability. So it would serve as a bacteriostatic, not necessarily a bactericidal. And then we have to keep in mind the spectrum for this antimicrobial treatment. A broad spectrum is going to be a chemical that targets something that's going to kill a lot of bacteria. Maybe it targets peptidoglycan. So it's going to kill your own bacteria as well. A narrow spectrum then is going to be one that targets a particular microbe. We talked about the mechanisms of action then. What do the antimicrobials do? We listed 5 things. They can interfere with the cell wall formation. They can disrupt the cell membrane, so punch holes into it. They can interfere with metabolic pathways. They can interfere with protein synthesis. They can interfere with nucleic acid synthesis. So anytime you're affecting a protein that's involved in any one of these pathways, or any one of these processes, you can interfere with the microbe's ability to reproduce. Nucleic acids. So the proteins, the metabolic pathways, the nucleic acids, the cell membrane, and the cell wall. Basically anything the cell does. If you can interfere with any of those vital functions, you can prevent the microbe from growing. And then, of course, bacteria are going to have mechanisms to resist these antimicrobials. We mentioned 4 mechanisms. It can decrease the uptake so that the microbe doesn't take up the chemical. Changes the gates that allow the chemical to come in. You know, maybe they had a channel this big and they shrink it down to this size. They can increase the elimination. So as the chemical comes into the cell, they can pump it out just as quickly. That's going to be energy intensive. It's going to cost a lot of energy to do so. They can inactivate the drug. If they produce a chemical that can bind to the drug so that the drug then can't bind to the target molecule, essentially removing the drug, preventing it from carrying out its action, or they can change the target molecule so that the shape is different. The drug now can no longer attach to it, doesn't recognize it. It's key that you understand this notion of acquisition of resistance, how bacteria become resistant to these chemicals. Mutation, of course, is the main means, but the simple fact that antibiotic drugs have been overprescribed and used incorrectly has led to resistance, development of resistance to a number of drugs. So, how are you going to go about studying for the diseases? Okay. The best thing I would think is to try to maybe put together some kind of chart for yourself, isolate or identifying the systems that they're in. So you got the respiratory system, the skin, the digestive system. Be sure that you know the viruses from the bacteria, because that can be key. I can give you a multiple choice question. Which one of these microbes is responsible for this condition? And if I'm telling you about which one of these is responsible for, let me think for a second here. Oh, I could say which one of these would microbes would this chemical be effective, or this antimicrobial be effective against? And I could list Yersinia pestis, and then I could list 3 viruses. Antimicrobials aren't effective against viruses, so you would recognize that it's not going to kill those viruses, and you would identify the bacterium. I could go the other way with that too. I could identify a disease and then say which one of these microbes causes this disease. If you identify that disease as being caused by a virus, even if you don't know the virus, you can eliminate those microbes that are bacterial, and by process of elimination, hopefully key on the microbe that causes the disease. Because you have to figure there's probably, 20 or more different diseases and microbes that we've introduced over the last 4 lectures. So there's a lot of stuff there. How I'm going to go about doing this is with matching in a lot of cases, where I'll give you several systems and you'll have to identify the disease that goes with that system, or I'll give you several diseases, you identify the microbes that cause those diseases. So there'll be some matching. But I'll also throw in there occasional true, false, and multiple choice questions with regards to the diseases as well. Give you a condition, and then give you several microbes, and you have to tell me which microbe is responsible for that condition. A couple of other things to keep in mind is the environments that these systems create, because that will tell you a lot about the types of microbes that are going to be there. So the skin, we've got a dry, arid, salty environment. You guys are doing your presentations. I don't know if you've recognized the fact that those of you who are saying you have Staphylococcus or Micrococcus, and you tell me that it grows in the high saline concentration. So that makes sense. If you indeed have microbes that are growing on the skin, then they're going to grow in those high saline concentrations. Respiratory is very different environment, warm and humid. What kind of microbes are you going to expect to find there? And then, they're going to have to get past these cells, the epithelial cells lining the digestive system, which is an easy route for a number of microbes. We see that with Shigella and Salmonella and whatnot. And then you have the digestive system, which again is a very in-hospital environment, you would think, except for the fact that there's a lot of microbes there. What's that?
No essay question.
No essay question. All multiple choice, true, false, and matching. One other thing to work on or keep in mind, we've focused on Staphylococcus aureus and Streptococcus pyogenes. So be sure that you study those 2 microbes. They both have several virulent factors that allow them to infect cells. So be sure that you understand how they are able to do that, what their defense mechanisms might be, and also the number of diseases and conditions that they cause. So I could list a disease and ask you which one of the microbes causes this, Streptococcus pyogenes or Staphylococcus aureus.
Can I ask a question on that real quick? So in regards to those 2, both of them cause impetigo.
Yes. Yeah. So, as you've seen, I could could set up a multiple choice where I have Streptococcus pyogenes is a choice, Staphylococcus aureus is a choice, neither is a choice, and both is a choice. Okay, so you would have to say, well, both of them have that ability, neither of them have that ability, something like that.
Are there any other ones that are crossed over like that, or is that a unique case where you have a condition or a skin condition that's caused by 2 different microbes?
No, there's a lot of...
Did we cover any other ones in here?
No.
That's the one we covered here.
Yeah, but there's a number of microbes that can cause similar conditions like that. Just like there are a number of microbes that can cause what is known as the stomach flu, a number of microbes that can cause, respiratory flu. You're generally just talking about symptoms in those cases that are similar, but the action The mechanism might be different.
So gastroenteritis, we talked about that.
That's the stomach flu, yeah.
Yeah, but I thought that was caused by viruses, P-virus.
That can be caused by viruses, can be caused by bacteria. It's just a gastroenteritis. It's just an inflammation of the intestinal system.
So it's viral or bacterial.
Yeah, exactly. Any other questions? So, I'm not going to talk anymore tonight. I'm kind of done. I'll be here Monday night. We can review again Monday night. I'd prefer to, instead of just me going over everything again, to actually address specific questions. So if you have questions, you need clarification about stuff, I'd like to do that. Also, there's a probably greater than 90% chance that I'll be in the laboratory on Friday at 11 o'clock for a little while. So if you wanted to stop by and talk about the laboratory material that we're going to be covering, I'd be happy to do a short review in laboratory Friday at about 11 o'clock. Okay. Be sure to get an evaluation form for 10 points.